So today I'm going to make a video about how to pour and run agarose gels, and we use this for gel electrophoresis of nucleic acids, either DNA or RNA. So if you'll see, I'm back at this back bench of the lab and by the chemical shelves, and this is where we're going to do our work for this activity. So the first thing I'm going to do is come over to I'm going to try not to make you too dizzy, come over to this sink, and there will be a few of these Erlenmeyer flasks available here. There's also a carboy right here with 1xTAE, and this is the running buffer and the buffer that will be used for pouring these gels. There's also a graduated cylinder here, and so we will use this to measure out the 1xTAE and to pour it into the flask. And today I'm going to run a gel using our smaller apparatus. So we have this orange apparatus here. We have a middle-sized gel running apparatus, and then over here, I guess you can see, there's a really large one. So for each one of your purposes, you want to count and make sure that you have enough wells for what you need to do. The orange apparatus, when we set it up, let's see if I can just orient you a little bit better here. When we set this up, we're going to flip over this container. This is going to contain our running buffer and our gel. We are going to place into this apparatus, we're going to place in this container, and whenever we run the gel, we're going to orient it this way so both sides of the gel will be exposed to the tanks of TAE. But when we pour the gel, we're going to put it in this way, and in that sense the orange walls will provide two of the walls of the gel, and these two plastic pieces in the insert will provide the other parts. So anyway, before you get your gel made, you can set up your apparatus. You're going to have this plastic piece inside the rig, and you can see that there are some little divots in the plastic, and that's where these combs are going to go. So the combs will fit in just like so. Before you decide which apparatus you're going to use and which gel you're going to use, you want to make sure that these combs have enough wells for your purposes. So if you had 100 samples, then this gel would not have enough wells for all of your samples, and you would need to change what kind of gel you're going to run. So this has on it, let's see if I can angle this for you. It says B1 to 14. That means there's 14 of these little wells on here. I have 20 samples, so if I have 28 wells, that'll be enough for all my samples and my ladder. So now I have set up my rig with my gel tray and my combs. One of the things that you can do wrong is to not have these combs set into the little spaces. And so if you see the side view, if there were one of these combs that was not in there properly, then your gel wouldn't pour correctly. You also want to make sure that this tray is snug. Sometimes you can wet it with a little bit of algal water if you need to. And you want to make sure that the rubber gasket is straight. So if this little rubber gasket, which is kind of orange, bumped up, then the gel would not pour correctly. So you want to check that for both sides. Okay. If you wanted to use this medium apparatus, which is right here, you would do the same thing. It also has a tray that fits in it like this, and it has these green combs that have 20 little wells, so it's a little bit bigger. And then the large apparatus, which most people don't use early on, but you can use eventually. It has a really, really huge tray that fits into this little container right here, and has these really long orangish red combs. So that would fit, and you can actually put many more of these combs in there. I think you can probably see there's a lot of these. So you could make 2 rows or 3 rows or even more if you had a ton of stitches. Okay. So because I only need 20, this is going to do just fine for me. And the next thing I'm going to do is make my agarose gel mixture. You'll need obviously a different volume of agarose gel for one of these types of gel apparatuses. Usually I make about 80 mils for this one, thereabouts. You can make about 100. I usually make 100 or a little over 100 for the medium, and then usually about 200 milliliters for the really large one. So what we're going to do next is combine our reagents to make the agarose gel. As I mentioned, the TAE buffer is in this carboy, and I'm going to measure it out with the graduated cylinder, put it in this flask. The other component is going to be agarose, and we have aliquots of this agarose, so it's easier to pour right here. And I'm going to weigh out some of this agarose on the scale here in one of these weigh boats. So let me go ahead and get that ready. So I'm going to put the weigh boat here. I'm going to hit T for tare so that it's zeroed. And then I'm going to measure out my agarose. Generally for nucleic acid gels, we want to make about a 1% gel. So that means if I'm going to use 100 milliliters, I'll have 1 gram of the agarose. If I wanted to make 80 milliliters, for example, I would then use 0.8 grams. So I'm going to measure out over here. Let's see if I can set this up a little better. I'm going to measure out 0.8 grams just by tapping this a little bit into the weigh boat. And this, I have a little bit over 0.8 grams, but this is now in my little weigh boat and ready to go. Okay. This is a little tricky to do on a video, so we're gonna do our best here. So now I'm gonna set you up with a little bit broader of a view of this part of the lab, so I can show you what I'm going to do. So I have a graduated cylinder. I'm going to fill it up to 80 milliliters with the TAE. I'm going to pour that 80 milliliters into this flask, just like so. And then I'm going to use my weigh boat with my agarose to pour the 0.8 grams into the flask. When I swirl it, you'll see that it's still white. That's because this agarose does not dissolve until it's heated up. So what I'm going to do next is come over to the microwave and put in the microwave. I'm putting it in for about a minute, and then I'm going to come back to it and show you what it looks like. So switch the view a little bit, but I'm over here by the microwave now, and I have my flask that's been microwaved for 1 minute. And you can see if I swirl it around, a lot of that white color has cleared out, but I think you can probably see that there are little orbs that are kind of a translucent color, kind of clear throughout the solution. So this doesn't yet look like water. So I'm going to put it back in the microwave and set the temperature for 30 more seconds, and I'm going to repeat this until when I swirl the solution, it's clear. The one thing you want to make sure you're careful about is that this can boil over. So after the first minute or so, I keep an eye on the solution through the door of the microwave, and if it looks like it's about to bubble over, I stop the timer and then give it a swirl, and if it needs a little bit more time, put it back for a little bit longer. And And this is what the liquid will look like. You can see here it's boiling and bubbling a little bit, so I'm going to hit stop and I will take the solution out. I'm using, I should have noted, I'm using one of these little rubber grips so that I don't burn myself. This will be quite hot. I give it a little bit of a stir. Be careful, if you stir it really aggressively and it's hot, it can boil over. And at this point, I don't see any more of those little agarose orbs. It looks as translucent as water. I don't see anything in it. You'll also notice I'm being a little bit careful not to introduce a lot of bubbles. So once the agarose solution looks like this, it is ready to proceed. So you can just close the microwave and stop. That for me took about 1.5 minutes. If you have a larger volume, it will take longer. So if we were to take this agarose solution that's boiling hot and to pour it directly into the gel apparatus, it would warp the plastic. So what I'm going to do next is to take this bottle over to the sink and run it under kind of cool or room temperature water to make sure that this is still warm but not hot. So I will kind of angle this like so. So I'm over at the sink. I'm going to turn this on with cold water and I'm going to swirl the I'm going to swirl the agarose so that it cools. I'm being careful not to get water in the agarose, but I'm just going to cool it down. I'm also trying really hard not to introduce a lot of bubbles. So I'm running the water down the side of the glass. And I'm also rotating the flask every so often so that it cools evenly. If you were to cool this down all the way to room temperature, you would find that the agarose solidifies. So what you're aiming for is something where, the agarose is still warm and still liquidy, but is not too hot. And so one of the ways that I kind of test this is to hold the flask with my hand. If I can hold it, that probably means it's cool enough to pour. If this is still too hot to touch, I would run it under the cold water a little bit longer. So if we were to pour this agarose gel without doing anything else, we wouldn't have any way to visualize the DNA. So the last thing we're going to add before we run the gel is a solution that's going to make the nucleic acid fluoresce under specific wavelengths of light. In our lab, we use something called gel red. So this is a little gel red. You use it at 1 to 10,000, so this is 10,000 times concentrated, which means that if you have 100 milliliters, you're going to use 10 microliters, or if you have 80 milliliters, you're going to use 8 microliters. This stuff is a really strong stain, so you want to be careful not to get this all over your clothes. But if you've used dyes like ethidium bromide before that are a little bit dangerous, this is actually pretty safe compared to those. So we like having this in the lab because we don't need to worry about our personal safety. So I'm pipetting 8 microliters of the gel red, and I'm going to just drop it into the agarose solution. I actually put the tip of my pipette right under the surface of the agarose to make sure that that droplet of gel red, which is not a very big volume, gets into the agarose. And if I show it to you, you might be able to see that it's still a little red drop in there. So I'm going to give it a little bit of a swirl to make sure that it's mixed. And then I'm going to carefully pour this agarose into the gel tray. I'm being very careful not to introduce a lot of bubbles. And if you did introduce any bubbles, you can do things like take the the comb out and put it back in if you notice there were bubbles next to your comb, for example. Make sure that they're in there carefully. And then you want to rinse out this flask with deionized water in the sink before it solidifies and makes little chunks of gel, and put that back on the drying rack where you found it. After pouring a gel, you're just going to want to make sure that you've cleaned up after yourself by putting away the agarose, putting away the little rubber grip and all the glassware that you used. You'll also want to make sure that you leave your gel be for a little while to make sure that the gel polymerizes, so that agarose, once it melts, will make a networked polymer once it cools down. So we will check on it in a little bit. And what you will see is that although this solution is very clear when you pour the gel, it will become somewhat opaque once the gel has polymerized. So I waited about half an hour for my gel to polymerize, and I've come back to check it. You can see that the gel is a little bit more opaque instead of clear. And if you look at it straight from the top, you don't see any differences in color. So if you saw some regions that looked kind of a darker color and lighter color, it might not be evenly polymerized. Once your gel's polymerized, you can go ahead and start setting up your samples to run your gel. So next I'm going to talk you through how to set up things to load your agarose gel. And in this example, I have hot start PCR reactions. And so if you have hot start PCR reactions, you can do things exactly the same as I'm doing, or either at the end of this video, I might include a few more videos for other situations like RNA or cloning PCRs, or I might have some text at the end to tell you how to do that differently. So first, I've taken out my PCRs from the PCR machine. I've also got a second PCR tube rack. And what I'm going to do is to cover part of that rack with parafilm. So parafilm is a kind of waxy film. There's a paper sheet on one side of it. So I'm actually going to cut a little piece of it off. And then sometimes they kind of stick together. Peel them apart. And I'm going to take this little wax piece and I'm going to put it over my PCR rack. And what I'm going to do is use my finger to press into this wax and make little tiny wells for each one of my samples. And the reason I do this instead of labeling tubes and things like that is just simply kind of speed and not using too many disposable items. So I like to just set up my reactions to load in one of these instead of going to all the trouble of labeling tubes. So now that I have my little setup ready, I have 20 tubes, so I have 20 little dents in my parafilm. I'm going to get things ready. So if I had just pipetted this straight into the gel, one problem I would have is that I couldn't see anything. And so what I'm actually going to do is combine some water and some loading dye and some of my samples and then get them ready to load. So the first thing I'm going to do is to add a little bit of water into each one of these little divots. The reason I'm going to do that is I actually only want to run out 1 microliter of this PCR. I don't want to waste it all on the gel. And because 1 microliter is hard to pipette, especially into a gel, I'm going to add some water as well. So what I'm going to do is add 7 microliters of sterile water, and I have the sterile water in a little 1.5 ml tube just to make it easier to pipette. I'm going to pipette 7 microliters of this using just one tip over and over into the little dents that I have made. Okay, so now I have 7 microliters of water. Just little droplets of water in each little dent. I'm trying not to mess them up. And the other thing I'm going to add is orange G. So orange G, there should be tubes of this around. I have a big tube of it somewhere on my bench. Let's see if I can find it. Not sure where it is. We have, oh, here it is. We have one big stock of this. But it has sucrose and an orange dye and water, and it's a really good loading dye because it's inexpensive and easy to see. So a loading dye is something that gives the samples that you're going to load two qualities. One is it'll be colored, either like blue or orange, so that you can see what you're pipetting. And the other thing is that usually loading dyes make the solution a little bit denser than water. That's what the sucrose is there for in this. So that the sample containing your PCR or riboprobe or whatever will sink into that well instead of just diffusing out into the water, in which case you wouldn't be able to run out your gel and see what you're doing. So I'm going to add just a little bit of this orange G to each well. You could pipette 1 microliter over and over again, but what I do is I just set my pipette for 10 microliters and I just add a little bit of that orange G to each. Well, I can get about 7, I guess, wells from 10 microliters. I'm just doing that for speed. So now each one of my samples has a little droplet of water that is orange. And then next I'm going to take my PCR samples and add 1 microliter of each one of my PCR samples to the same spot in my little setup here. So what you don't want to do is just mix them up, right? You need to know that what you think is in sample number 1 is in sample number 1 and so on. So I'm going to take my first 8 samples and put them on my top row, samples 1 through 8, and my second row into samples 9 through 16 and so on. So I'll do one of them and then I'll pause the video so you don't have to watch the whole thing. So I'm just going to take 1 microliter of each PCR, being careful not to drop this. So 1 microliter, just a tiny little bit, and I'm going to pipette this into the little orange droplet that's for sample 1. And now I'm going to pause the video, but I'm going to do that for the rest of my 20 samples. Okay, so now I've made a little group of samples here. You can see them all orange. I've put up my PCRs into the fridge because I don't want them to be out at room temperature while I'm loading my gel and running my gel. And now I'm going to take my 10 microliter pipette and very carefully take my tray with my samples to the back bench where my gel is. Actually, take this phone stand too. Okay, just be careful not to drop this. You'll have to redo what you've set up. I'm just going to put this pipette and my samples right next to where my gel is set up. The other thing I'm going to get while I'm getting ready to set up is I'm going over to the fridge. So that's this VWR fridge right here. And I'm going to get out something called Hyperladder 1KB. So it looks like this. It has kind of a whitish lid. And this is a ladder, which is a tube containing lots of different nucleic acids that are specific links, like how many base pairs long they are, so that you can compare your bands to bands of known size. So if you know that your insert or your PCR product should be a certain size, you can use the ladder to determine whether that is so. Okay, so here I am with my gel rig. The first thing I'm going to do is to get this gel ready to go, and I'm actually gonna get a better angle on this. I'm going to first take out my combs very carefully. I kind of brace my hands with my middle fingers and just gently pull this comb out. If you pull it too quickly, you can rip your gel. So I'm going to do that for both of my combs. And now you'll see that the gel is polymerized and it has these little holes in it for your samples. The next thing I need to do is gently take this tray out so that I can rotate it. And I'm going to rotate it so that it is long ways in the rig, like this. I'm actually going to do it the other way. So the wells are at the top and in the middle, and these electrodes are on the right hand side. Okay, and the next thing I need to do is fill this apparatus up with the 1X TAE. So I'm going to come over here to the carboy and add buffer. You can also fill up a big graduated cylinder and do it that way. And making this TAE is a lab job. It's someone's lab job. So if it looks like we're about to run out, you can let someone know that we need to make more. Okay. So what I'm going to do is load this gel. I'm actually going to try to turn the camera around so that you can see what I'm doing a little bit more easily. And I'll show you exactly how to load the gel. What I'm going to do is load 3 microliters of the loading dye. Then I'm going to skip a well to give a little extra space, and then I'm going to pipette all of my samples after that. So I've never tried to do this entirely one-handed, but we're going to give it a go. What I need to do is I need to set my pipette to 3 microliters. Your pipette might look different from this. And get a pipette tip. And then I have the loading dye set up here. And I'm just going to pipette 3 microliters of that. And then I'm going to angle myself so that I can see what I'm doing. Some people like to be kind of at an angle. Some people like to be right above. Some people like to put something under the gel, like maybe a paper towel, so that they can see the wells better. So you can kind of experiment with what you need to do. There you can see that's a little bit better. And then I'm going to pipette into this sample right here. What I typically do when I'm not filming myself is I will use my left hand to brace my pipetting hand to give myself a little bit more stability. And that's just because you may notice that your hand shakes a little bit, and using your left hand to brace your right hand or vice versa will help with the shaking. Okay, so now I'm going to pipette my other samples. So I have another pipette tip, and I'm going to use that to suck up this first sample. It should be about 10 microliters. It might be a little bit less, and if so, that's okay. And then I'm going to come over here, and as I said, I'm going to skip a well, and I'm going to pipette this into the next well. What you should see is that that solution should sink into the well and stay there, so you can see the ladder that's purple and then the sample that's orange. If you see the sample diffuse, you may not have gotten that tip actually down into the well. So if you see here, I don't have anything in my pipette, but I'm going to put the tip into that well. You can tell it's in there because I can wiggle the gel around a little bit. You don't want to jab it all the way to the bottom because you can pierce the bottom of the gel, but you do want to be pipetting into the well. If you pipette some liquid just onto the surface, you know, if you were to just pipette right above the well, it may not all go in there. So what you should see is that each one of your samples should sink down into the well, and then you should be able to see them. So I'm going to load the rest of this gel and then come back. So as soon as you're finished, you can return the ladder to the refrigerator. I also wanted to just kind of show you how this looks when I'm doing this two-handed. If I'm using two hands, then I will use my left hand, I'm right-handed, to kind of touch this part of the pipette with my middle finger and brace my left hand on the side of the gel apparatus. And this gives me kind of some stability when I'm trying to angle my pipette where it needs to go. And so you can kind of experiment with that, which way is easier for you. And this will take you a while. I'm pretty fast at this point. I've probably pipetted thousands or tens of thousands of wells of gels at this point, but it's okay that it will take a little while the first time. And if you want to grab someone when you're doing it the first time just to make sure you're doing it right, that's fine. You can also discard all of your tips. You want to use a fresh tip for each sample. You don't want to get them mixed up. And then what it should look like when you're finished is you should have ladders on each line of samples, and then you can see the samples out beside it. What you're going to do next is add the lid. So we have lids like this. These lids have connections. And you're just going to put this on the top and slide it over so that it looks like this. Let's see. You want to make sure that these electrodes are connected like this. And then you're going to plug in the ends of these leads into the power box, which is right behind where this setup is. And it's color coded so that if you have the black electrode at the top and the red electrode at the bottom, then you can plug them in the color coded way and it's correct. So DNA is negatively charged. It's going to run toward the cathode or the red end. And so I always in my head have the mnemonic run to red. So if you have it set up properly over here, the red will be positive and the DNA will run toward that red end. I'm going to turn on this unit, so let's just flip you around. Turn it on by touching this middle button. It's going to give you a voltage, so you want to run this at 100 volts. You can, sorry about that, 100 volts. You can use this to select the time, so I'm only going to do 45 minutes and then I can do longer if I need to. And then I'm going to hit run. And once you hit run, what you should see is some bubbles rising up. I'm not sure if you can see them in the phone, but you should see some bubbles right at the end of the gel, the foot of the gel, and at the top of the gel. Okay, so my gel has run, and I can tell that because my apparatus says off. I'm going to press power, and make sure that the bands that come from the orange G or your loading die have moved from the wells into the gel. If they've done so, then your gel has run properly. You can then lift out this tray and then be really careful not to drop your gel. You can kind of use a thumb to make sure your gel stays on the tray. And then you can bring this over here. I've opened the door since I'm doing this one-handed, and you can place your gel here. You can either remove this from the tray or put the whole tray here to image the gel. Then you're going to close this door, and if the oxygen program is not open, you can go ahead and push this button. If it is open, you're going to select dark room, and this will allow you to image your gel. This might look a little weird because it's a screen. You're going to use UV light to excite the fluorescent molecule that's in that DNA dye, gel red, and you can see that what's come up is my ladder and then my PCRs. You can also see some numbers. This is just from some fluorescence on the tray that I put in there. It wouldn't show up if I had taken the gel off the tray. If your gel doesn't show up quite like this, you can change the exposure by either moving this little bar around. If the bands look way too bright, I would decrease the exposure. If you don't see anything, increase the exposure. If you still couldn't see anything, just make sure that you have the UV setting correct, that you have a long enough exposure time, and make sure that you added that gel red. If you don't add the gel red, you might not see what you're expecting. So then you can capture the image. This will take a few seconds if you have a long exposure time. You can then do things like crop. So let's see, if this is kind of hard to do with one hand, but you could crop your gel however you want it, and hit crop. And then you can print. And I would say cancel. The other thing we often do is invert this so that it shows up as dark bands on a white background. It just looks a little nicer when we print. When we print, I would say you go to preferences and select multiple pages per slide and then do like maybe 6. The reason for this is if you don't do that, you will print a gel image that's an entire page big, which uses a lot of ink and doesn't fit into your lab notebook very well. So you can go over to the printer to make sure that your printout came out, and if so, you can just X out of this program. You don't need to save your gel image unless it's something important for a publication. So I made sure that my printout came out of the printer. I'm then going to dispose of this gel by carefully just taking this over to the trash can. Because we have a non-toxic dye, we can just do that. And then I'm going to use a little bit of algal water to spray off the surface, and I'm going to get some chem wipes to wipe up that water. You want to leave this clean. Again, you can just X out of the program and not save your image if you don't need it. And then after this, you can move on by doing the next step in whatever protocol you're working on. Just quickly, I forgot one thing of the cleanup, which is you need to put away your materials. You can unplug the lid that you used and return it to the shelf. There's usually some space around here. You're going to take both your tray and your gel running apparatus and rinse them out in the sink with deionized water. And the only reason, if you were going to run another gel right after this, you could reuse the TAE once or twice with no problems. But if we have plenty of TAE, and if you don't need to reuse it, you can just go ahead and rinse these out and return them either this way or face over to dry.